Good afternoon, everyone, and welcome to MSK Science Spotlight. My name is Thomas Tamala. I'm an assistant member in the Cancer Biology and Genetics program at the Sloan Kettering Institute. This seminar series, which has been running now for a while, features lectures from today's leaders in basic and translational biomedical science. If you have questions throughout the talk, you don't have to wait until the end. You can actually email them directly to the Office of Scientific Education and Training at osed@mskcc.org or by tweeting to at MSK Education. And these will be shown here on the slide. And then questions will be answered at the end of the program. For the best viewing experience, use Google Chrome. If you're using Internet Explorer, please use version 11 or higher. If you experience technical issues during the live stream, please lower the video resolution. You can do that by clicking on the gear shift on the lower right of the screen. And now to the star of the show. I am pleased to welcome Dr. Alexandra Joyner. She's a member in MSK, in Sloan Kettering Institute's Developmental Biology program. And her talk today is titled, Constructing the Brain, Ensuring Plasticity and Repair. Please, Alexandra.
Thank you, Thomas, for the introduction and for all of you for tuning in here. I know these have been pretty trying times and quite difficult, especially these last few weeks, perhaps for the United States more than the rest of the world. However, I'm starting to feel hopeful that some of the inequities in Western societies related to race, health, care, and wealth actually will perhaps start getting addressed. So let's just be hopeful. For those of you who don't know me, I'm a developmental biologist. However, I actually am one of the very few neuroscientists at Sloan Kettering Institute. But I hope you'll stick with me, even if you might feel a little daunted by the brain. And hopefully you'll see how our work has, in basic neural development, have implications for certainly a lot of work being done in the Brain Tumor Center, as well as elsewhere around the Institute. Sorry, it's not moving forward. I have to restart it again. Let's see if it's going to work now. Okay, good. So at heart, I'm a developmental biologist, but over the years, I followed the science, and it really took me more and more into the nervous system, which I'm quite grateful for, because the brain is quite a fascinating area of the body to study. Very complicated, but very interesting. But I thought it would be interesting to go back to my first paper as a postdoc with Gail Martin, soon after she had cloned or discovered embryonic stem cells, and look back at what we were planning to do. And it's really what I'm still doing. And that was, we hope to identify genes in mammals that controlled morphogenesis, so how structures are formed in 3 dimensions during development, by taking the newly cloned Drosophila genes and looking for mouse homologs. And that turned out to be extremely effective. And of course, most of you probably heard of the Hox genes, which are transcription factors that control many aspects of development. But we cloned 2 related homeobox genes called the Engraved genes. And we're looking for genes these genes were of interest because they were known to control segmentation or border formation, or as we would call it now, lineage restriction in fly, and we're hoping for the same kind of genes in mouse. And nicely for us, when sorry, it's not going forward again. I have to escape and start again. I hope it's not going to keep doing this. when I started my lab, a student early on made antibodies and was able to look at the expression of these two genes during embryonic development. And we saw this great expression in this segmented pattern down the somites, which form the vertebrae, this interesting expression in the limbs, which once we were able to use our signature fate mapping to mark these cells in the early embryo and look to see what they become, we saw that this gene nicely marks the ventral ectoderm, and phenotype analysis with Cindy Loomis showed that Engraved was critical for defining the ventral ectoderm. And in its absence, the ventral ectoderm takes on a dorsal phenotype such that even the nails are duplicated. And of course, they lose the sweat glands. But what we ended up deciding to focus our work on was this region of the brain, which we came to understand marks the entire midbrain and the anterior segment of the hindbrain, called rhombomere 1, which gives rise to the cerebellum. And the reason for focusing on the cerebellum, I think you can see nicely here in this movie of the developing brain for the first 2 weeks after birth. And you can see that the cerebellum forms these beautiful folds. And I thought it could be a fascinating area that wasn't being studied to try to understand how these distinct folds are formed, because every single mouse has the same folding pattern, and human has a somewhat distinct, but also a very highly conserved folding pattern. The other nice thing, not being a neuroscientist, was the cerebellum only has a few cell types, which I'll show you in a minute. And over the years, we have tried to understand this morphogenesis problem, but today I actually want to focus on some more recent work where we've been trying to look at the question of how during development are all the different cell types scaled properly so that they're balanced properly. You have the right amount of excitatory and inhibitory activity to make a functioning circuit. And then most recently, we ended up looking at the question of repair. What happens if you damage some of those cell types? Can they replenish themselves? And will the cells stay coordinated in this scaling process? So you can see the cerebellum has only a small number of cell types. There's just 2 cell types that project to the cerebellum. They both project to these gigantic cells called the Purkinje cells, either directly or indirectly by these granule cells, which are the most numerous cell type in the entire brain and actually body. The Purkinje cells then project down to the cerebellar nuclei, which is the smallest number of cells in the cerebellum, and they're the output neurons that talk to the rest of the brain. And the question we were interested in is how do the right proportions of these just different neuron types get defined during development. And in the developing cerebellum, there are some extra cell types, of course, because we need the progenitors. And these green progenitors, I'll talk about in the second half of my talk, and they give rise to the interneurons and the astrocytes. But I'd be remiss not to mention this other cell type called the granule cell progenitors, because they are actually the cell of origin of the brain tumor sonic hedgehog medulloblastoma. And since moving to Sloan Kettering 15 years ago, we have been studying this tumor in trying to understand cell intrinsic and extrinsic regulation of both initiation and progression of the tumor, but that's a story for another time. So why study the cerebellum beyond it has this beautiful folded structure that as a developmental biologist I'm intrigued by. It also is a critical part of the brain as it basically enhances all the functions of the cerebral cortex. So not only movement, which is most usually equated with the cerebellum, but also cognitive, language, and social behavior. So it's a very critical part of the brain. And you can see the brain is actually made up of almost many mini organs. Each of these brain regions has 6 to 12 different cell types. They all have their own progenitor domain. So the brain is actually put together, I would argue, by a number of smaller organs, although that have to talk to each other to work together to make the circuits function. And one interesting question, since the circuits actually go across the entire brain as a developmental biologist question is, are the cells in the cerebellum aware of the cells in the rest of its circuit during development to know how to proportion properly, not only within the cerebellum, but relative to the rest of the circuit. And that also then brings up the question during evolution, when new circuits were established, how does that occur when you have to produce cells in all these different regions? And as I'll show you, we think part of it is utilizing survival factors, that the cells in these different regions all depend on survival factors. And so if you increase one cell type, other cell types can then be stabilized or survive and allow new circuits to form. And I think you'll hopefully understand a little better as I get into the first part of my talk. So when we first knocked out these two Engraved genes after I'd started my lab, we found they had quite distinct phenotypes. The Engraved 1 mutants die at birth because most of the cerebellum and midbrain is gone. And this reflects its very earliest expression in the part of the neural tube before it even closed, where this region, the midbrain and the cerebellum, are going to form. Engraved 2, in contrast, those mutants live, although they do have a smaller cerebellum. And most interesting for us, and part of what brought us into the cerebellum, was that these mutants have a change in their folding pattern. So instead of having these 5 folds, they only have 3 folds, and it's actually these 2 posterior folds that become fused. So a very specific change in the pattern of the folding, and we wanted to understand that. And the other thing we discovered, that by developing the knock-in approach to replace Engraved 1 coding sequence with the Engraved 2, was these 2 proteins are actually very similar. And so these distinct phenotypes are mainly because their gene expression is different, not because the function of the proteins is different. And that's meant that all the studies we've continued on have used conditional mutants of both genes because they have these overlapping functions. And what I want to first tell you about is trying to understand why there are only certain folds decreased or missing in Engraved 1 mutants, and how that led us to this question of scaling. And this work has all been spearheaded really by Sumru Bayan, who took over from a previous postdoc, Ryan Willett, and 2 terrific graduate students from the neuroscience program, Andrew Lee and Anjana Krishna. And, but I must first just give you some understanding of where and when the different cell types in the cerebellum are generated, because that's been a part of the whole story to understand that before we could even understand the Engraved mutants. So like all of the brain, the inside of the neural tube is lined by a group of cells called the ventricular zone progenitors. These stem cells give rise to all the neurons and astrocytes or glia. But in the cerebellum, the ventricular zone actually only gives rise to the inhibitory neurons and astrocytes. In contrast, there's this little structure at the back of the cerebellum that gives called the rhombic lip, that gives rise to all the excitatory neurons. And unique to the cerebellum, they actually migrate over the cerebellum. And first they produce the cerebellar nuclei, these output neurons, and then they make a progenitor zone on the outside of the cerebellum with these precursors of the granule cells. At the same time as this is happening, the ventricular zone produces these gigantic Purkinje cells, and then it produces another progenitor, again, that's unique to the cerebellum, because it leaves the ventricular zone and keeps proliferating out in the what we call the cerebellar cortex as these lobules are growing out. And these nestin expressing progenitors, or NEPs, gives rise to the interneurons and astrocytes that are all born after birth. And during development, these 2 the cell types given rise by these two progenitors actually exchange positions, so that ultimately the granule cells are on the inside of the cerebellar cortex, the interneurons on the outside, and sandwiched in between are the Purkinje cells. So what ensures that the granule cells and interneurons and astrocytes are proportioned in the right numbers? Well, what our work and others have shown is that Purkinje cells express sonic hedgehog, that important morphogen, but in this case is actually acting as a mitogen and as a molecule that keeps the NEPs and the GCPs in an undifferentiated proliferative state. And so before they generate the postmitotic cells. So they signal to these two proliferating progenitors and regulate their expansion. And so in a sense, the amount or the concentration of sonic hedgehog then scales the production of granule cells, interneurons, and actually astrocytes. And by extrapolation, then it's the number of Purkinje cells present in the cerebellum at birth that determines the growth and the scaling of all these different cell types. And I have to mention that ATO1 is a basic helix transcription factor expressed only in these excitatory lineages as the cells leave the rhombic lip, as it's a ATO1 Cree mouse that we've used a lot in our work. So what we've seen in all the mutants that we make conditional or null that are viable is that they always have a smaller cerebellum, and usually one set of folds, but different folds depending on the mutant, are preferentially reduced. At the same time, and this is using ATO-Cree to just knock the Engraved genes out only in the excitatory neurons. At the same time, what we see is the cytoarchitecture of the cerebellum looks very normal, indicating scaling is actually preserved in these mutants. And we recently, we being Andrew and Anjana and Sumru, painstakingly quantified all the different cell types in these mutants, and we saw that they were indeed scaled properly. So this would be the reduction in just the area of the cerebellum. The Purkinje cells, which are not affected by this Cree, are also reduced, but their density is normal, so their scaling looks normal. The interneurons, which also are not affected by this Cree, also have a normal scaling, as well as the granule cells, even though they are mutant. And the ratios of the different cell types are preserved. The one other main cell type in the cerebellum, the cerebellar nuclei, they come actually as 3 pairs rather than a layered structure. I mean, 3 pairs of nuclei. And we found, interestingly, they are reduced, but they're only reduced in the medial and the intermediate nuclei and not in the lateral nuclei. And this is just showing you quantification across from medial to lateral. It's normal once you get lateral. And that made us think, well, what about the areas in these different regions? And we saw that indeed the areas only reduce in the medial and the region called the vermis and the intermediate region called the paravermis, but not out in the hemispheres, indicating a relationship between the number of these cerebellar nuclei and the growth. And indeed, when we look further and looked at where do the Purkinje cells from these preferentially reduced lobules project, they actually preferentially project to the part of this cerebellar nucleus that is reduced, indicating again some causative perhaps relationship between the number of CN and the growth of the different lobules. So 2 other things to note is in these mutants, what we found is the cerebellar nuclei are initially born in their normal numbers, but then die by embryonic day 17, so just before birth. And the Purkinje cells also are born in a normal number, and then start dying after this, we believe, after the CN die. So why are the Purkinje cells numbers reduced in these mutants? And one problem is, and what determines the number of Purkinje cells even normally present at birth. And I'll show you our model, and then I'll show you some of the evidence for it. And it's based on a theory that's been around for a long time called the Neurotrophin Theory, that mainly seen in the peripheral nervous system, and that is that neurons produce a neurotrophic survival factor that's required for the survival of their presynaptic neurons. So for the CN, their presynaptic neurons are the Purkinje cells. And the idea is if in the Engraved mutants these neurons die, then the Purkinje cells will die because they don't have their survival factor. And then the growth of the expansion of the granule cell layer will be toned down because there's less Purkinje cells, less sonic hedgehog, but everything should still scale normally to this reduced number of Purkinje cells. And that's, in fact, what we believe happens in these Engraved mutants. One problem is, though, that in these mutants, we're knocking out both the granule cells as well as the cerebellar nuclei. So we had to devise a method to separate the knockouts into the two cell types. And we were able to utilize an ATO-TTA allele transgene we'd made to turn on a Cree that would then knock out the Engraved genes. And as you probably know, you can feed animals doxycycline to inhibit TTA activity. And since these cell types are born at different times, depending on when we give doxycycline, we either knock the genes out in the cerebellar nuclei or in the granule cells. And indeed, what we found, it was only when we knocked out the Engraved genes in the cerebellar nuclei that the number of CN were reduced, and again, specifically in the medial and intermediate. And again, the cerebellum vermis was also reduced only in these mutants, interestingly, not in the granule cells. The granule cells, the Engraved genes seem to play only a minor role in these cells in terms, at least, of growth. And again, only in the vermis and the paravermis and not in the hemispheres. Arguing further that there is some kind of causal relationship between the number of cerebellar nuclei and the growth of the lobules that their presynaptic Purkinje cell neurons are a part of. And so to finally put the nail in the coffin in this, what we did was to kill a specific subset of cerebellar nuclei and ask, does that result in death of the Purkinje cells and smaller growth of the cerebellar lobules or the folds? And to do this, we developed a new transgene that might be useful to some labs, where the attenuated diphtheria toxin protein is expressed only in cells that have both expression of TTA, so we use the ATO-TTA, and that have Cree to delete this tomato stop signal. And by using the appropriate Cree, then what we were able to do was kill 50% of the cerebellar nuclei, but now equally across the cerebellum, including out in the hemispheres. And indeed, what we see then is growth of the cerebellum is reduced in all three regions, and the Purkinje cells are reduced and do then die after the CN are killed. But again, the scaling is maintained. And so in summary, what we were able to find using this Engraved mutants, but to find about normal development and scaling, is that in these mutants where you only specifically have a reduction in these medial and intermediate CN, then you get a reduction in growth of only the paravermis and the vermis, and even more so this preferential loss, because a subset of the very specific subset of the medial CN is lost. We get this patterning defect. So it all is a domino effect from these small number of cerebellar nuclei, the first neurons to be born, end up affecting the growth and patterning of these folds. And of course, if we kill CN throughout the cerebellum, then the whole cerebellum just reduces in size but would have normal patterning. And in terms of what I brought up before, how these, does development of the different regions of the brain, is it interdependent? What we're interested in now is if we specifically kill some of the targets of the cerebellar nuclei, like the thalamus, will we get a reduction of their specific presynaptic CN neurons, and then that should result in the rest of the domino effect, so that the Purkinje cells are reduced and growth of those specific globules connected to the thalamus would be reduced. So I want to now, from the rest of the talk, switch to a different question, and that has to do with the ability of the cerebellum to repair itself during development. And this is important because, as I showed you, the cerebellum primarily grows after birth, and that actually means that the cerebellum is actually particularly sensitive to any kind of injury at birth, and in particular, premature birth often can result in cerebellar injury. And it's been known then also that cerebellar injury is the second leading risk factor for autism. So it would be really important then to understand the degree to which the cerebellum is plastic enough during development to undergo repair to any injury that occurs at birth. And then this would help because the cerebellum affects so many different behaviors. We could try to eventually develop approaches to even enhance regeneration. And this work was all started by a former postdoc, Alex, who then was joined by Sumru, who's carried on the work. And Alex was interested in what if we, given we know there's this kind of scaling going on, what would happen if we kill particular cell types around birth? Can they be regenerated? And if so, do they undergo normal developmental processes, or can we uncover new processes? And the latter, of course, is what we found, since I'm going to tell you about it. So Sumru, after she arrived, she decided to kill the Purkinje cells around birth and ask whether they could be regenerated, which I just have to admit thought was a crazy idea. Purkinje cells, they've been born a week earlier, you can't get proliferation and new neurons, but in fact I was grossly wrong. And she was able to show that you can get an actually 100% regeneration of these Purkinje if she kills about 50% of the Purkinje cells at birth. And this is just a really pretty picture showing that where the red is the dying Purkinje cells and the green are the cells that survived or have been regenerated and that are now able to push growth of the lobules. And what she discovered was that there's a dormant immature Purkinje cell that we didn't know exists that comes from the ventricular zone, probably at the same embryonic day 10 to 13 time point. And it, in response to death of its neighbors, actually can proliferate one or two more times and regenerate those Purkinje cells that have died. And this process, though, is very age dependent. So already by postnatal day 5, these dormant immature Purkinje cells are greatly decreased in number, and regeneration is very poor, and the animals end up having actually behavior defects. What Alex had started with was instead killing the granule cell progenitors, and since they're a proliferative progenitor, we kind of expected they could regenerate. But the really surprising finding that Alex found was that these nestin progenitors that normally only give rise to interneurons and astrocytes, they actually contribute to the regeneration of these granule cells. And we get up to about 80% regeneration if we kill these granule cell precursors at postnatal day 1. And this is what I want to focus on the rest of the talk, is how these cells undergo adaptive reprogramming to become granule cell precursors. sorry, I meant to miss this slide. So what we knew when we started this project based on fate mapping by us and others was that there seemed to be at least two groups of these nestin progenitors, NEPs, nestin expressing progenitors, one in the Purkinje cell, or what we call the Bergmann glial layer, that gives rise to Bergmann glia, these specialized glia, and astrocytes, and the other in the white matter that gives rise to interneurons and also thought to give rise to astrocytes. And if we kill these granule cells, and we most prevalently just use irradiation, which I'll show you, what happens is this cells specifically in the Bergmann glial layer respond to this injury by proliferating, migrating out to the EGL, and then changing their fate, turning off nestin, turning on ATO1, and becoming granule cells, progenitors, and making granule cells. At the same time, interestingly, this group in the white matter also can sense this injury, and they actually slow down their production of interneurons and astrocytes, like they're waiting until the granule cell layer is replenished and granule cells start to be made again. And as you might expect, sonic hedgehog, Alex showed, is required for this regenerative process. But we were interested in understanding this at a deeper level than just the sonic hedgehog. And just to show you a shout out to Pat in the small imaging core, it's been tremendously helpful for us that we can target just the hindbrain, midbrain of these postnatal day 1 pups. And you can see 1 day later, high tunnel labeling specifically in the granule layer, external granule cell layer, and fortunately not in the layers where the NEPs are, likely because they divide much less and much slower. And just in case you think it might all be due to irradiation, we've also used genetic killing specifically of the GCPs in various ways, including diphtheria toxin, and get exactly the same results as what I'll show you. And I think the most convincing evidence for this migration, of course, comes from live imaging, something Kat here has really been the pioneer in, and Andrew Lawton got going in our lab when he was a postdoc. He's now at Mississippi State, and he took slices of the cerebellum, put them into culture, and then imaged them for about 6 hours. And we were fortunate to have this nestin expressing, nestin CFP transgene that we can visualize live very easily. And you can see in the normal cerebellum, there's really very little movement of these progenitors. But if you look after irradiation, so this mouse was irradiated at P1 and the slices are taken at P6, you can see there's already nestin cells in the external granule cell layer. And moreover, if you watch the ones in the Bergmann glial layer, you'll see some migrate very rapidly once they get going from the Bergmann glial layer out to the external granule cell layer. So what stimulates them to these NEPs to change their fate and go out to the external granule cell layer? And this is what Sumru has been trying to track down for the last couple of years, so far focusing on what are the transcription factors that allow these granule cells to do this fate change. And to do this, you might not be surprised, we turned to single cell sequencing. And what she did was take the whole cerebellum at postnatal day 5 of mice, either non-irradiated or irradiated at postnatal day 1. And postnatal day 5 is the stage when everything's happening: proliferation, migration, and reprogramming. And this was a fantastic collaboration with Peter Sims' lab at Columbia University with Do Mizrak, his postdoc, using their microwell platform. So all the major analysis, single cell analysis, is done by them. And here's just shown all the data from the 16,000 cerebellar cells put together. Here you can, and we got all the expected neuronal groups and associated cell types. And here you can see by condition that we do get changes after irradiation. They're not totally on top of each other. Each condition. And importantly, when we look for the nestin CFP transcript, it's enriched in this one cluster, which we would believe would then therefore include the NEPs. And fortunately, 67% of these cells express this, or we can detect the transgene in a high percentage of the cells. But there's also, interestingly, a number of CFP cells in these other clustered. And so what they decided to do was just take out the nestin CFP or the CFP transcript expressing cells and focus on those cells and ask, can we identify biologically relevant subpopulations and then identify transcription factors under controlling this adaptive reprogramming. And so this shows you the two conditions mixed together of CFP positive cells. We now get, interestingly, a number of different subpopulations, not just one. And then perhaps more useful, when we separate them, we saw that in the wild type, there weren't just two flavors of NEPs, but there's at least 4 different ones. But only one of them has a high CFP, and as I'll show you, turns out to be the Bergmann glial layer one. The other 3 actually all seem to be NEPs in the white matter, and so they're a more complicated subgroup. And nicely, after radiation, we still get this same group, but we get 2 new groups. This pink group that I'll focus on that we believe has come from the Bergmann glial layer, and additional white matter groups. Now, if we just first start by focusing on the wild type condition, what we see is I'll show you the markers that identify these different groups. There's GDF1, 10, and SLC and HOPX are interesting genes, all you can see enriched by RNA in situ from Allen Brain Institute in the Bergmann glial layer. And so there are nice markers for this. The white matter NEPs have been harder to find markers, but we've got 2 genes that are enriched in all 3 groups, and you can see they're not enriched, barely expressed in the Bergmann glial layer. And then interestingly, we were able to identify one of these white matter groups as expressing neurogenic genes. So we believe it's a neurogenic group in the white matter that's the precursor of the interneurons. And fortunately, we had made a CRE-ER for this ASCL1 CRE-ER gene and could use it to fate map those cells. And then we were able to get in a HOPX CRE-ER to fate map the NEPs in the Bergmann glial layer. And it confirmed what we thought, that you can see 2 days after tamoxifen at P5, the time when we did the single cell seq, most of the cells that are labeled with ASCL1 are in the white matter. And the few cells that are in the Bergmann glial layer, we believe are actually migrating cells, because these cells, while they're born in the white matter, they migrate out into the molecular layer. And indeed, when we look at in the adult, virtually every cell marked becomes an interneuron in this molecular layer. In contrast, the HOPX CRE-ER mark cells in this Bergmann glial layer, and more interestingly, these cells have the glial structure of Bergmann glia. And this glial structure is important during development for granule cells. And you can see out in the adult, fate mapping this, these give rise to only Bergmann glia, as well as astrocytes, and interestingly, also a small percentage of interneurons. And so this little group here was a little, we hadn't predicted initially, but there seems to be this interesting HOPX positive progenitor also in the white matter that presumably gives rise to some of these interneurons and possibly some astrocytes. But this Bergmann glial structure of these stem cells then in the Bergmann glial layer indicates then that they have the structure of a Bergmann glial cell, and which is normally used for these granule cells to migrate down into the IGL. So we went from thinking there were two different net populations in the developing cerebellums to seeing that there's at least 4 different ones, 3 in the white matter. The one in the Bergmann glial layer expresses HOP-X and gives rise primarily to Bergmann glia and astrocytes. There's also a subgroup in the white matter that's neurogenic and gives rise to interneurons exclusively. And there seem to be, we haven't identified the markers for these astrocyte producing progenitors. But we have found, and I won't show you, that they're actually can be separated based on the HOP-X. It only makes astrocytes that are found in the lobules, whereas there must be another progenitor for the astrocytes down in the deep white matter, which is where the cerebellar nuclei are, and of course are more lateral than in the cerebellum. And the fact that we see HOP-X giving rise to some interneurons makes us wonder if there isn't even earlier upstream progenitor that's bipotent that gives rise to both the astrocytes and the interneurons in the lobules. So what I want to end with then is just showing you what happens if we irradiate the cerebellum, that what we believe happens is this HOP-X progenitor in the Bergmann glial layer now gives rise to ASCL1 expressing progenitor that then produces the Bergmann glia. So we can see here that if we look at the single cell seq from the irradiated mice, of course there's this Bergmann glial layer markers are expressed, but interestingly this new cluster actually expresses some of these markers, actually the more immature markers, and these markers don't change their pattern, so they're still restricted to that Bergmann glial layer. The white matter markers, however, are reduced, both the general white matter marker as well as these neurogenic markers. And that fits with what I said at the beginning, that these white matter progenitors actually dial down their production of interneurons and astrocytes at P5 during regeneration. However, this ASCL1 now comes on strongly in this new cluster that basically has some properties like the Bergmann glial layer and some like a neurogenic cluster. And so we propose that this is the lineage then of this new progenitor. And I've been able to use fate mapping to prove that. So if we use our HOP-X CRE-ER and give tamoxifen at P0, irradiation a day later, and then look at P7, and what we see is these new cells that are induced in the Bergmann glial layer with the HOP-X actually express ASCL1, whereas the ones that are normally marked do not. And here's just the quantification. And if we fate map those out to the adult, what we see, again, if we quantify, there's these new cells generated by the HOP-X labeled progenitors, specifically the granule cells in this IGL. So this HOP-X cells give rise to ASCL1 expressing cells, and those also produce granule cells. So we can perform the same thing with the ASCL1 CRE-ER after irradiation and ask, do those give rise to granule cells? And indeed, that's what you see if we fate map to P30. Now we have to give tamoxifen at P5 when ASCL1 is highest. And of course, in the normal animal, it's a simple lineage, just giving rise to interneurons. After radiation, presumably this lineage is still there, but now we have this new lineage that's giving rise to granule cell progenitors quantified here in the internal granule layer, but we also unexpectedly found that the cells also produce astrocytes. But interestingly, no Bergmann glia. So once the cell expresses ASCL1, it does not go back to making Bergman glia. And here's showing you, if you look just 2 days after tamoxifen, these cells that are marked in the Bergman glial layer do indeed have this Bergman glial fiber-like structure, whereas normally you just have, as I showed you before, these migrating interneurons. So the final question you're probably asking is ASCL1 actually required for this fate change to reprogram this interneuron making cell, or this Bergman glial making cell into making granule cell progenitors? And so just recently, after coming back from the shutdown, Sumru's been able to look at her first set of animals where she used this HOPX-CRER then to knock out ASCL1 in these Bergman glial layer progenitors by giving tamoxifen at P0, irradiating a day later, and then looking at the adult. And you can see that in the normal animal, you have a reduction in the size of, or the area of the cerebellum. But if you also now knock out ASCL1, you get much reduced or greatly impaired regeneration. And even at the cytoarchitecture level, this is a marker for the granule cells. You can see the IGL is quite disorganized. So we've kind of followed the science, as it were, and gone from trying to understand morphology to finding out that, you know, the firstborn cells in the cerebellum actually set the domino going to for the rest of the cerebellum to grow and be patterned properly. And then the cerebellum has this amazing ability to regenerate. And in this case, we found the importance of ASCL1 in inducing this adaptive reprogramming of these NEP cells. So here is just the summary then of all these different cell types that we found, and that after injury, then this ASCL1 expressing progenitor or intermediate progenitor transient cell is induced that can now make granule cells. And it also makes a small number of astrocytes. Curiously, these astrocytes actually go into both the deep white matter and in the lobules, which makes us start to wonder, then makes me start to wonder if there's actually two different progenitors here, one that makes astrocytes and one that makes Bergman glia, and perhaps it's this astrocyte progenitor that actually becomes the ASCL1 cell and undergoes this adaptive reprogramming. So just want to end by thanking my group, highlight Andrew, Sumru, and Daniel. Anjana, that generated most of the data I showed you, and Daniel and Jimmy, who've been tremendous help, long-term technicians. So I think that we need a new name for them. Lab managers running the lab for now for more than 20 years, and very fortunate. And also like to thank my lab for the amazing resilience over the last few months. This is from our first Zoom meeting back in March, but you can see now that they're back in the lab, we're all much happier and enjoying generating the new data. So I'll stop there and take any questions.
Thanks, Alex, for a really wonderful talk. Beautiful biology, very cool approaches. There are several questions that came up. I'd like to start with one of my own. Just a very sort of general question. What is the role of synaptic connections in this process? Do they regulate Engraved or hedgehog or ASCL expression? What is the sequence of events there?
That's something we really haven't addressed specifically at this point. It's a really interesting question. Definitely, we believe a lot of it's kind of cell intrinsic developmental processes, but definitely also we're coming to appreciate that the talking between neurons, and possibly even at the level of the synapse, rather than just BDNF being a survival factor, may well also be playing into this whole thing. And there are genetic tools now we could use to, and actually Andrew is kind of interested in that idea of trying to dampen down activity and see if that alters growth or patterning.
That sounds great. And maybe also some
a former postdoc who's got his own lab has done that a little bit. It didn't have a major impact on the overall growth morphology, but we haven't looked in detail.
Great. Okay, then 2 questions from the audience. So the first one is a sort of another fairly general question. These survival factors that you've talked about for cerebellar circuit formation, are they only found in the cerebellum, and are they similar perhaps in other parts of the brain or other organs?
Yeah, they definitely play a role in the peripheral nervous system. They're not really as well recognized in the brain, but I am guessing now we're going to find that that's actually true probably throughout the brain. In the cerebellum, we know BDNF is expressed at the right time in the cerebellar nuclei neurons, but it alone doesn't seem to be sufficient. So I'm assuming there's several of them expressed, and no, they won't be unique to the cerebellum.
Okay. And then does the cerebellum differ in its structure in humans and non-human primates from the mouse?
Yeah, I didn't show that picture. They actually have the same basic major folds. It's quite amazing. In the human and the mouse, they actually even label them the same. But of course, the human, instead of having one large lobule like the mouse, has 30 smaller lobules, and then even these tiny little folds on top of that. So it has a tremendous surface area compared to the mouse. But the basic circuits also have been pretty well preserved. So certain lobules regulate motor behavior, other ones social, and those actually seem to be relatively well conserved between mouse and human.
Okay. Then there's a question about the scaling process and the repair. So the window seems pretty narrow in the postnatal stage. Do you think there would be ways to extend that window, perhaps?
Well, that's actually what Sumru's really interested in, and got a K99 to hopefully in the next year or so set up her own lab to really push that further. She has been able to show that there are NEPs, or nestin CFP expressing cells in the adult cerebellum. They do respond to injury in that we get an increase in the number of them. We also can increase them with sonic hedgehog, but they really do very little in response to injury in terms of repair, producing new neurons. So she's now wants to compare them, the early postnatal and the adult, to see if we can see transcriptionally, using ATAC-seq and single-cell-seq, what's different and what might be inhibiting the adult ones from being able to be more plastic and start proliferating and do something really useful.
That sounds really interesting. And actually a follow-up question or a related question to that pertains to your comments about autism and this idea that in the mouse model also, if you ablate these cells, mice develop behavioral phenotypes. So would that suggest that injury has a connection to human autism as well? Perhaps autistic children have had this type of injury in a critical stage of their development. Is that what you think?
Well, that's what, that graph I showed, because the second highest risk factor anyway for autism is known injury to the cerebellum. Okay. Developmental injury. So yes, and I didn't mention this, but Andrew's done a whole set of behavior analysis on those Engraved mutants I showed you, and they actually do have some social interaction defects. So he's trying to track down that neural circuit out to the cerebral cortex and see what's altered in the Engraved mutants.
Got it. So another question regarding the repair response has to do with inflammation. So do you think inflammation has a role in repair from injury, and perhaps immune cells as well?
So something we're just starting to look into Sumra and another postdoc and in the lab. We've started from just looking at the potential effect of just altering ROS, which ends up altering all kinds of things, and it does actually seem to be from a transgene affecting regeneration, and we can see early on the microglia moving out to the EGL during that, but it's very transient actually. We've only seen it like one day after irradiation during the actual cell death. They're very quickly cleared away, but we're very interested because of course they usually are involved in regeneration. The thing you have to remember though, at that time, everything's very immature. The microglia are much more immature than normal. And these Bergmann glia and astrocytes, most of the astrocytes haven't even been formed yet. So some of the cell types are either not there or are immature compared to in adult injury. So it's going to be a complicated project, but I think it's interesting. Okay.
There's a question about the engraved transcription factors themselves. You've very nicely shown their role in critical function, dissecting genetic experiments and spatial temporally, but what's known about their targets? And could some of those targets actually be potentially used therapeutically?
Yeah, no, very good question. And that's what Anjana, another student, is one of her projects. We actually made a HA tagged Engraved 2 mouse line recently, so that we can start to do that because we have limited polyclonal antibody. And so that's one of the things. The problem, the challenge is that the cerebellar nuclei cells, if we want to look there, are the smallest. There's only about 10 or 20,000 per brain. So it's a bit challenging, but she's now got ways to sort the cells and pull them out. So we're hoping. And we may also look in Purkinje cells where we know Engraved's playing also a role, and there's tenfold more of those cells. And start to discover what are their targets, because obviously, that's what we really ultimately want to know.
Okay, maybe one final question. This is for me. I'm interested in the HOPX positive cells, which seem like a new, previously undescribed cell type. So what do you think? Progenitor cells obviously have the potential to transform. So what do you think of their potential role in as cells of origin for perhaps medulloblastoma?
Yeah, I didn't mention that, but I'm predicting that they will be the cell of origin of one of the other types, type 3 or type 4, because there's another Wnt group that's already been shown to come from the hindbrain, actually not come from the cerebellum. But my guess is either group 3 or group 4, which are more driven by either NMEC or CMEC, are a cell of origin potentially of those. But we just didn't have a good genetic way to model that at this point. But I'm sure somebody will at some point.
What's known about HOPX expression in those types of medulloblastoma?
Good question. We'll have to look. Good question. That's easy enough to do online very quickly. Well, I don't know if we've done that because it's kind of the whole HOPX thing came up more recently than when we were kind of thinking about the broader questions. But our fate mapping and stuff also say it says it's a subset, although it seems like it may be that preferential cell type that is involved in this adaptive reprogramming. So it is a special cell. And there is one group, Lee's group, that had shown, at least in the embryo, that the Bergmann glia express HOPX, but nobody's been looking at it after birth.
Okay, very interesting. It's 5:30, so we're right on time. Thanks, Alex, for a really wonderful presentation. I enjoyed it, and I'm sure the audience did as well. So I want to thank you and our audience for joining us. Our next seminar is happening this Wednesday, on June 24th. And then we're pleased to welcome Jed Walchuk. And the title of his lecture will be Enabling Co-Stimulatory Pathways as a Next Step for Cancer Immunotherapy. So I hope to see many of you next week. Thank you, and bye-bye.
Everybody. Bye.